Okay. Good day, everyone. I'm Meiqi Niu from Nicola Palomero-Gallagher's group in Research Center Jülich as a postdoc. And since part of our major work is to make a receptor-driven, comprehensive architectonic brain map of the macaque monkey. So today I would like to show you an example to illustrate what can we do with it when we have such a precise, multimodal, and informative brain atlas. So today I would like to use macaque somatosensory region as a carrier to explore the relationship between receptor and functional connectivity pattern. To help individuals interact accurately with surrounding stimulus, the somatosensory systems receive sensations detected in the periphery and then transfer those informations via the pathway through the spinal cord to brainstem, thalamus, and literally to the cerebral cortex. In the cerebral cortex, the information is still generally processed in a hierarchical way from the primary to the secondary somatosensory areas, and then finally to the association cortex. Although there are lots of experiments provide evidence for this kind of hierarchical processing, there are still lots of questions remain poorly understood. For example, we don't know how are these local hierarchies organized throughout the entire somatosensory system, and whether the different hierarchical level of the functional system can be reflected by their structural characteristics. And to answer these questions, we must understand how information flows in the entire somatosensory network. And the brain is essentially an intricate network of connected neurons. And since receptors are the key molecules that enable signal transmission between different nerve cells, the well-toned balance between multiple receptor types could reflect the chemoarchitectonic mechanism of this network. As such, exploring the similarity of multiple receptor distribution pattern of area in question could be a proper way to link the brain structural and the functional connectivity at the system level. So in the present talk, we aim to establish the receptor and functional connectivity pattern, and then compare their similarities and differences. Specifically, we first define the region of interest based on our own architectonic parcellation, and the seed areas include 34 somatosensory related areas in the parietal lobe, and target areas include 125 areas covering the cortical surface except for the temporal lobe. And then for each area, we first calculated or extract 14 different receptor type densities by means of the quantitative in vitro receptor autoradiography to make a representative feature vector. At the same time, we also measure the representative activity time course for each area. And the resting state MR data used here stemmed from the prime DE dataset. And then for both receptor and resting state MR data, the statistical similarity between two brain areas were calculated by computing the Pearson correlation. So here you can see the group interregional correlation matrices. To facilitate the description and interpretation of our result, the seed area was divided into 5 groups based on their hierarchical levels or anatomical locations. And as you can see here, the visual inspection indicates that the anatomically adjacent areas share numerous similarities in their receptor covariance patterns. And this the receptor covariance patterns are also similar for those areas which are at the same or similar hierarchical levels or belonging to the same subsystems. For example, we found all primary somatosensory areas. They showed consistent correlation with anterior part of the intraparietal sulcus, inferior parietal lobe, and higher visual areas V3, V6, and part of the anterior part of the prefrontal area 46. While the areas within intraparietal sulcus, they showed consistent correlation with entire superior parietal lobe and posterior part of inferior parietal lobe, most of the visual areas, as well as prefrontal area 11. On the other hand, we also found the areas belonging to the same subsystem can also be separated by their receptor covariance pattern. So for example, within the primary somatosensory cortex, we found the areas 3BLI and 3BLE. They could be separated by, from other S1 areas, since they have relative higher correlation with most of the visual areas, especially the primary visual areas, but they also show lower correlation with anterior inferior parietal lobe, intraparietal sulcus, as well as part of the prefrontal areas. Then let's move on to the next question. What are the similarities and the differences between receptor and functional connectivity patterns? Regarding the similarity, not surprisingly, almost all areas showed consistent, strong correlation with their surrounding areas in both RC and FC. And as to the differences, the most obvious difference is that RC connection strength is higher than that of FC from an overall perspective, although RC also shows more negative correlation. And the difference is also reflected by the connectivity pattern between somatosensory areas with other cortical areas, such as motor visual areas, as well as medial and lateral part of prefrontal cortex. And the final point of the differences is that if we compare the connectivity pattern among all somatosensory related areas, we will find that they have similar FC patterns, but variable RC pattern. As to the mechanism for these differences, we suppose that FC reflect the degree to which different brain regions or networks exhibit synchronized or coordinated activity over time. That means areas simultaneously activated by certain tasks will cluster together. So this can help us identify the functional system. While the receptor covariance patterns reflect the similarity in multiple receptor balance and between different brain regions, so it allowed us to estimate the potential for brain areas to be responsive to similar neurotransmitter receptors or signaling molecules. That is to say, if two brain areas they have similar receptor architecture, it could suggest that they have similar hierarchical levels or perform the similar roles within a specific functional network. And this is also in line with our previous observation. We found the primary sensory, primary visual areas, they have relative unique receptor fingerprints since they receive less information from other cortical areas. But the association areas have relative general and similar receptor fingerprints since they exchange information more intensively. And as the work I showed today is just a part of our preliminary results, so of course there are lots of future work are needed. First of all, obviously it's very important to include the receptor data of the temporal lobe, and some of our students are starting the postulation and the density extraction work of this part, so I believe it will deepen our understanding once we fill this gap. In addition, as you already know from the first talk in this session, our previous colleague, Thomas Funk, he's building the first normative atlas of the neurotransmitter receptor distribution of both human and the monkey brain. So that means we have chance to further explore this question at the works level. I believe this method will undoubtedly help us improve the accuracy of our receptor covariance patterns. And in the end, neither FC nor RC imply the accurate or implies the direct anatomical connectivity. So we are expect to explore the anatomical connectivity by using the same brain atlas, and I think this will help us understand the relationship among multiple modality of connectivity and how they interact to each other. So I would like to thank all my colleagues and collaborators for their support, and of course, thank you for your attention. I'm open for any questions.
So we are open for questions. Yes, please.
Amazing work.
Thank you.
I just wanted to ask how you were going to do the anatomical connection stuff.
I would like to do, but I don't have the, by now I don't have the proper idea to use what kind of data. In my own point of view, probably it's better to use invasive tracer tracking data, but I also believe the neuroimage data will help us to address this issue.
Okay, very cool. And just follow up really quickly, the temporal structures being excluded, is that because like something about the brain, and so you're going to need a different brain to look at that, or is it just a matter of like?
Just because we need a long time to work with it, so we're just in the process.
Yeah,
but
sense.
Definitely, whenever we fill this gap, yes, we can do that.
Very cool, amazing work.
So you had mentioned in future work incorporating association areas, the temporal association. We had heard earlier today about moving into the allocortical areas, limbic, entorhinal, and so on. Do you already have that, or are you thinking of expanding there, or?
I think for entorhinal cortex, we didn't have the fine version of pacillation, but some of our colleagues, they worked on entorhinal cortex for right and human. Maybe Nicola can answer this question.
Piriform, yeah, of course it's difficult.
Well, you're infringing on my talk, maybe, which will be later. There's a PhD student I've got working right now on hippocampus and entorhinal cortex on this thing here. Yeah. And there's another PhD student working on the lateral temporal cortex, and there's a postdoc working on the auditory cortex. And I've got a PhD student that's just finished the amygdala, so it's work in progress.
Oh, okay, and so for either of you, what about laterality differences? Left-right hemisphere?
Sorry, what differences?
Laterality differences.
What do you mean?
Left-right hemisphere of the different areas.
Okay, because what we worked is monkey brain, is a macaque monkey, so actually the difference between left and right hemisphere, if I understand your answer, your question correctly.
Yeah, but you might be in a position to identify them more so than
we hope we can identify, but if just we look at by the visual inspection, we can see that the difference between left and right hemisphere is not that big in at least the macaque monkey brain. But we also compare it with human. For human, there are big differences in between left and right hemisphere.